In this video, I am going to show you how to perform 16S microbial analysis with nanopore data. What we are going to do is to first download the data that we are going to use, and then we are going to perform quality control and data improvements. After that, we will perform taxonomic classification on the data, and then we are going to visualize the results. This video is based on a metagenomics tutorial that I saw on the Aggregate platform. This is the page, and I will leave the link to this page in the description box. And so in this tutorial, we are going to use the command line versions of the tools that were used here. So take note of that. I'll later make a tutorial where I show you how to use the Aggregate platform to also do this analysis. So that's about it. In order for you to follow this tutorial, you need to make sure you have a Linux or macOS system. And you should also have Anaconda installed. We are going to use Anaconda to install the tools, which are here. But if you have the tools already installed, then you don't need to have Anaconda on your system. That's what you need to know. Instructions for installing the tools are in the description box. So I have tutorials that show how to install these tools. Most of them I've covered them. So check the description box for links to those videos. So now that we are done with the introduction, let's start the exercise. So let's open our terminal and then start the exercise. After opening our terminal, we need to install the tools. We are going to use Anaconda to do this. So make sure you have installed Anaconda. Again, instructions for installing Anaconda can be found in the description box. After installing Anaconda, what we have to do is to add channels. We will add 2 channels. So I'll say conda config add channels, and I'll say Bioconda. This is the first channel. Let's add a second channel. So the same command, but this time we change the channel name to conda forge. We have added the 2 channels. Let's list the channels. So you can say conda config show channels. This will list the channels. So we should see the 2 channels we added. The default channel comes packaged with conda, so we will leave it as it is. We are now going to create an environment and install the tools in that environment. So I'll say conda create-n, and I'll say 16s. This will be the name of the environment. Now we are going to list the tools. So I have fastqc, I have multiqc, I have fastp, I also have chop, I have KRAKEN 2, I have krona. So these are the tools. So we are going to use fastqc and multiqc for quality control. Fastp will be used to filter the reads. Chop will be used for trimming the reads. KRAKEN 2 will be used for taxonomic classification, and then krona will be used for visualization. So that's what we have. So now let's run this command to install the tools. Now let's type Y to confirm, and then hit the enter key. Once we do that, Anaconda will download all the necessary packages, install, and configure them for us. So let's wait for Anaconda to finish its job. Okay, the tools have been installed. If you use Anaconda to install the tools, then it's likely you will see these messages. I am going to focus on just the first paragraph here. If you're an advanced user, then you can take a look at the ones here and then make modifications, but I'll focus on just this paragraph. So I highly recommend you also do same. Now let's look at what has been displayed for us. So there are 2 things here. We are being told that we need to manually update the taxonomy databases that Krona uses to generate taxonomic reports. And number 2, we need to take note of the default location for storing the taxonomy databases, which is here. So that is what we have. So we are going to update the databases. But before we do that, let's also take note of what has been displayed for us. Okay, so there's the default location for storing taxonomy databases. So make sure you keep this somewhere. Yours is likely to be different. So whatever has been displayed for you, make sure you save it somewhere. Just take note of it. So for me, I'll copy it and then I'll place it in a text file so that I can always open that and then use that information there. So that's what we are going to. So we are going to update the database using the script, and then we will proceed. So first of all, let's activate the environment that we use for this tutorial. So I will clear the screen and then just go and then update the database. So first of all, I am going to activate my conda environment. So I'll say conda activate 16s. And then I am going to create a working directory and then I'll cd into it. So I'll say make the 16s. And then I'll cd into it. So cd 16s. And now I'm going to open a text file and then place in the default location for the krona databases. So I will say nano krona db.txt. And then I'm going to save that part there. Okay, I'm doing this so that in case I forget it, I can just display that text file and then just see what I have. So I'm going to save this. So I'll do an ls and then I'll see the file. So I can always do a cut to display the parts and then I can always use that. So make sure you also do same. Okay. So now what we are going to do is to update the krona databases. So I'll say kt update taxonomy.sh. So I'll run this command to update the databases. Okay, so the databases have been updated. So now we are good to go. So let's proceed. So what we are going to do next is to download the example data. So let's go back to the Galaxy page and then look at the download links. Okay, so if you're on the Galaxy page, just scroll down and then when you get to this section, you can click on Get Data. But before you do that, you can also read about the data. So there's background on data, so you can read about the data, but I'll skip that. So I'll move straight to Get Data to download the data. So I'll click it and I'll be sent to this section. So here, I'm going to see the download links for the file that I'll use. So what I'll do is to copy these files. I want to do a batch download, so I'll copy the files. I'll copy. So what I'm going to do is to copy the download links. Okay, so these are the links for the file. So I'll copy them because I want to do a batch download. So I'll highlight them like this and then I'll copy. But alternatively, you can also right click each of them, like open in a new tab or open link, and then the files will be downloaded for you. So you can do that one by one to download the files. Okay, you can do, but I would like to do a batch download, so I will copy everything here and then I'll go to my terminal. So I'll go to my terminal and then create a bash script, and in that script I will enter the links and then I will download the file. So let's go to the terminal. So on the terminal, I'll create a bash script. So I'll say nano. So that is nano. So I'm using the nano command to open a file. Okay, you can use any text editor, VI or any text editor for that matter, but I prefer to use nano. So nano, and then I'll specify a name. So I'll say download data.sh, and then I'll enter my shebang line. Then I'm going to paste the links. I'm going to use wget to download the file. So what I'll do is to issue wget at the beginning of each line, like this. And then that'll be it. So wget, and then I also supply it with the links. Let me just remove the extra space here. So this is it. So wget, and then I have the link. So I'm going to use wget to download all the files here. So this is my script. So I will close this file and then save the changes. I'll also do this. I'll do an ls just to check the file. So this is the file, the script. So I'll now add execution right. So I'll say chmod plus x, and then I'll specify the file name like this. So now I'll do an ls again, and depending on the Linux system you are using or the Unix system you are using, depending on the system you are using, you are going to see the color of the file change. So that's it. So now I'm going to execute this command to download the file. So I'll say /download data.sh, and then I'll execute it. So the files will be downloaded for me. So let's wait for the files to download. So here, you don't need to write a script for it. The most important thing here is to download the files. So whether you use a browser or you write a script, just make sure you get the files. That is what is important here. Okay, the files have been downloaded. So let's clear the screen and then check them. So you can do an ls, and we are going to find the files here. That's what you have. Okay. We are now going to create a directory and then move these files into that directory. These are fastq files. So I'll say make the data, and then I'll move the files. So I'll say mv star.fastq.gz, and then I'll specify the directory. So I'll move them there. So now let's do an ls. You will see a directory called data, this one here. Let's do an ls into it, and we are going to find a file there. Okay, so we are just trying to organize our files. Okay, so whenever you are performing an analysis, you need to make sure that you have directories that will be used to store certain types of data. So that's what we have here. Okay, now we have that done. So what we are going to do next is to perform quality control. So we are going to run fastqc to perform some analysis, and then we are going to use multiqc to aggregate the results, and then we can view using our browser. So that's what we are going to do next. So we are going to say fastqc, and then we will say data/star.fastq.gz. So let's run this command. Okay, we can see fastqc has started performing the analysis. So it will do the analysis for all the files. So let's wait for fastqc to complete its work. Okay, fastqc has completed its work. So let's check the output file. So let's do an ls into data, and we are going to see some files here. So for each fastq file, we will have an HTML file and a zip file. So that's it. So we have them. So that means we are going to have 4 pairs of files. So we have HTML and zip for each of the fastq files. Okay, now we have 4 files here, or let's just say 4 samples. So if you have lots of files, if you run fastqc on lots of files, okay, then it can be cumbersome trying to open the reports one by one. Okay, the reports can be found here in the HTML file here. Okay, it can be cumbersome trying to open them one by one using your browser and then looking at the results. So this is where multiqc comes in. So multiqc can be used to aggregate fastqc reports. So we are going to aggregate everything, and then we will generate a single report, and we can view that report using our browser. So this can help us to easily compare the files that we want, or the files of interest. So to aggregate the fastqc report using multiqc, we will say multiqc, and then we say data/star_fastqc star. I'll do this, and then I'll execute the command. So multiqc will automatically detect all the reports and then aggregate them for us. So we have that done. So it says it has found 4 reports and it has aggregated all of them. So we can find the output here. We have the reports and then the data. So these are the 2 files. So we can check them out. So let's do an ls, and we are going to find this file here, this HTML file, and then this directory. So we are going to view this one here. This has all the reports. So let's do that using our browser. Okay, so this is the multiqc reports. If you want to check the individual reports, you can find them in the data directory. So you can find them here. There are 4 of them. We are going to use the multiqc report because it has all the reports. So let's open this file using our browser. Okay, the report has been opened. So you can scroll through and then check the plots. Explanation for this report can be found on the Galaxy tutorial page. So when you are here, just go to the quality control section. So just check that section and then you find explanations here. Okay, so we have some plots here and then we have some statements here explaining what these plots are. So I will encourage you to read the document here, check the explanation, and this will help you to get a better understanding of your data or of the data because we are using the data here. And you can transfer the knowledge to your data as well. So it's just a matter of applying what you have learned. What we are going to do next is to improve the data set quality. We are going to use 2 tools, PORCHOP and then fastp. PORCHOP will be used to remove adapters and chimera, and then fastp will be used to filter the sequences. So with PORCHOP, we are using PORCHOP because this is nanopore data, and PORCHOP is one of the tools that is used to remove adapters and chimera from nanopore data. The reason why we are removing adapters is that adapters can interfere with alignment of reads to 16S RNA gene reference database. And then the chimera also needs to be removed because they can result in artificial inflation of the microbial diversity. So the information can be found here. Okay, the reasons why you have to remove adapters and chimera can be found here. So just read this statement and you get them. We are also going to filter the reads because this will help us to increase the specificity of the analysis. That has also been stated here. So just read the information here to understand or to know the reasons why we are performing some of these activities. So try and then read the document here. Okay, so we will start by using PORCHOP to remove the adapters and then chimera. So let's go back to the terminal and then continue. Okay, we are back to the terminal now. Let's take a look at our files. Let's do an ls into the data directory. We have some files here. Let me modify this command. So I'll say lsdata/star.fastq.gz just to display the fastq files. So we can see them here. There are 4 of them. So we are going to use PORCHOP to remove adapters and chimera from these files. So let's go to the PORCHOP documentation page to look at the example command we'll use for this exercise. So this is the PORCHOP GitHub page. I will leave the link to this page in the description box. So when you scroll down, you'll find the table of contents. So let's look at the quick usage examples, and then we'll see some example commands here. We are going to use this one here, the basic adapter trimming. Okay, that's what we are going to use. So I'm trying to reproduce what was given here on the Galaxy tutorial page. So that's why I'm using the basic adapter trimming. Now let's take a look again at our files. From the ls command, we can see there are 4 files. And so if you have 4 files, then you have to automate the process. Sometimes you can have hundreds of files. And so it will be better if you write a script to process the files instead of trying to write a command for each of the files. Okay, so I've already prepared a script that we can use to process these files and remove the adapters and then chimera. So let's go back to the GitHub repo. I already have the script prepared on one of my GitHub repos. So let's go to that GitHub repo and then get the scripts. Okay, so this is the GitHub repo which has the scripts. So this is my Metagenomics GitHub repo. I'll leave the link to this page in the description box. So we are going to use the script trim read.sh. So you can download or you can just copy the commands here and then use that. So let's open this script, trim_read.sh. So click it and then it will be open for you here. So this is my shebang line which I use. Okay, so that we can use bash as the default program to open the script or to run the script. And then I also have make that trim. So because we are going to remove adapters and chimera, we need to place the output in another file. That is what I do. So the make that will create a directory for me, and then I'll issue my porchop commands to process the files. So I have 4 commands here because I have 4 files here. So I first call porchop and I specify -t4. -t is used to specify the number of threads. I am using 4, but you can change it. You can use 8, and depend on the computational power available to you. And I also have -i to indicate the input file, and that can be found here. And then I also have -o to specify the output file. So the outputs will be saved or stored in the trim directory. So that's why I have trimmed, and then I also have this one here. You also notice that the input file and then the output file, the base names are the same. We have bulk bottom and we also have bulk bottom. But then when we come to the trimmed files, I have .trimmed. I have a naming convention, so I use names to also describe the files. So .trimmed here will let me know that these are files that have been trimmed. So that's why I have that here. And I also have -- format to indicate the format of the file. So the output file, what format should it be? We are using fastq.gz. You can specify other formats as well. So just go to the porchop documentation page and you find all the information that you want. So this is the command. So this command will be run, okay, and the same will be applied to the other files. So that is why I have 4 lines here. So 1 command for each file. If you have lots of samples, then this approach may be cumbersome. There are ways to kind of write and automate scripts, and that can be used to process several files. So if you have lots of files and then you have a challenge trying to process them, you can always reach out to me and then we can work something out. My contact is in the description box. Okay, so let's go. So that's what you are going to do. I am going to copy all the commands here and then I'll paste them in another script. So basically what I'm going to do is that I'm going to open a file on my terminal and then I'll copy and paste this command there. But you can also download the scripts. Okay, so that one is up to you. If you want to download, just come to click the raw. Okay, again, let me just go back. If you want to download, click on the raw button here. Then you can right click and then go to save pages. You can do that. But I will want to just copy the text because it's not much. So I'll just copy them. And then I'll move to the terminal. So on the terminal, I'll open a file. I'll say nano trim_reads.sh. You can use any text editor, your choice, VNI, but I prefer nano. So I just use what you want. So I'll open it and then I'll paste the text there. So that is it. So I have my command. So I'll now save the changes and then I'll add execution rights. So I'll say chmod plus x, and then I'll specify the file name here. So that has been done. You can do an ls to check the file. So this is it. So now I'm going to execute this command. So I'll say // and then I'll indicate the file name. So let's execute the scripts to process the files. Okay, so PaulChop has started this work. So let's wait for PaulChop to process all the files. Okay, the adapter and camera removal process has been completed. So you can see some text here telling you what has been done. Okay, so we'll proceed. Let's clear the screen and take a look at the files that have been generated. So let's do an ls, and we'll see that a directory called trimmed has been created. This directory contains the output files. So let's ls into it. So we'll say ls trimmed. And we are going to find our files here. So we will proceed. So what we are going to do next is to filter the reads. So these files will be our inputs. We are going to use fastp. So if you want to read more about the fastp tool, you can visit the developers GitHub page. Let's go and take a look. This is the page, and I'll leave the link to this page in the description box. So when you scroll down, you have some information here which will be very helpful. So it does lots of activities. Okay, it's comprehensive. That's what I'll say. So you can install using Bioconda. We've already done that. We also have example commands that you can use. So it can be used to trim adapters. It can also be used for quality filtering. You can use it to filter based on lengths. There are lots of activities here that you can do, okay, using fastp. So I wouldn't go into details. I just wanted to show you. So let's go to one of my GitHub repos. We are using the Metagenomics repo. So I've prepared a script that we can use to filter the reads. So the script is here, this one, filter_reads. So it has the fastp commands that we'll use. So let's open it and then look at what we have. So I have that here. So I have this command to create a directory, and I also have the fastp commands here. Okay, and I also have this one here, this those lines that begin with hash. So these are comments telling you what each of these flags or options mean. So that's what we are. So I'm going to copy the commands here from here up to this one. I'll copy them. And by the way, before I copy, let me say that the options here, I selected them because that was what was used in this Scholastic tutorial. So if you take a look at the filter sequences section, you have the options here. So I try to reproduce what has been given here. So just take note of that. So let's go back. So let's copy our commands here, and then we will go and then paste them in a script that will generate. Alternatively, you can download the script itself, but I just thought I'll do it this way so that I'll use that to explain some of the codes here. Okay, so let's go back to the terminal. So on the terminal, I'll create a script. So I'll say nano filter reads.sh, and then I'm going to paste the commands there. These are the commands. I think I omitted this one here. Let me add it here, the shebang line. Okay, so I'll exit and save the changes. So I am now going to add execution rights. So I'll say chmod plus x, then I'll call the file name, which is filter reads. I can do an ls to check the file. It's here. Okay, so now I am going to execute the scripts. So I'll say // filter reads, and then I'll execute it. Okay, so fastp has also done its work. So let's take a look at the outputs. So let's do an ls and we'll find another directory called filtered reads. So let's ls into it and we are going to find our filtered files or filtered sequences. So you notice I also have this one .filt. Okay, so it indicates that these ones have been filtered. So that's how we do it. Okay, we have completed our adapter and camera removal steps. So let's go back to the Aggregate page and look at what we are supposed to do next. So when you are here, scroll down to this section, reevaluate dataset quality. This is what we are going to do next. So here, we have to analyze the files that we generated, the outputs from the filter sequences with fastp. And the data that was generated here, we need to analyze them using fastqc and multiqc. But I'm going to skip this step because the focus of this tutorial is to show you how to assign taxonomic classifications. But I will encourage you to reproduce this step and then read what has been stated here. This will help you to understand the data in the tutorial. So just make sure to do that. But in general, if you run the fastqc and then the multiqc commands, you are going to see something like this. So this here tells us that the anomalies in the sequences have been successfully eliminated. Okay, so if you compare this to the original data, you see some differences. So we corrected some of the issues, okay, using the trimming, that's the adapter remover, camera remover, and the filtering of the sequence. So that's what we've basically done. So in other words, we have improved the data. So the reevaluate data set quality is just to help us help us to compare what we have done and to make sure that we've really improved the data. So that's what it's about. So just make sure to reproduce it and then just continue. So I'll move on to the next step, and that is to assign taxonomic classifications. And we are going to use KRAKEN2 to do this. So whenever you are dealing with metagenomic data, it's important to identify the taxon that the individual reads belong to, because you may be dealing with several microbial organisms. So it's important to know where each of these organisms belong to, okay, with respect to the taxonomy. So that's why we have to do this. So we are going to use KRAKEN2. And by the way, I have covered another tutorial on KRAKEN2. So if you're interested, you can watch that tutorial. The link to that video is in the description box. I also encourage you to check the developers GitHub page, KRAKEN2. I will encourage you to check the developers GitHub page to read more about KRAKEN2. So you can visit this page. I will leave the link to this page in the description box. Also check the manual, which is here. I will leave the link to this page in the description box as well. So make sure you read these documents. It's going to be helpful to you. So let's go back to the Aggregate page. So we are going to run KRAKEN2 and we are going to reproduce what has been given here. Now, if you are going to use KRAKEN2 to assign taxonomic labels, you need to first get a database that you will use. So if you look at this tutorial, they use Silva. So we are also going to download the Silva 16s database and then use that for our activity. So we are going to do that now. I have prepared two scripts. One will be used to download the 16s Silva database, and the other will be used for the taxonomic classification task. So that's what we are going to do next. So we have our files, which are here. We are using these files, okay, because these are the ones that we have removed the adapters, camera, and we have also filtered them. So these are the final file that we are going to use. So what we are going to do next is to download and set up the 16s Silva database. So let's first go and get the script that we'll use for this exercise. So the script we will use is this one here. And again, I will leave the link to this GitHub repo in the description box. So first, let's click this script. Let's click it, and then we will have it displayed for us. We will have the content in it being displayed for us. So that's what we have. So I'll explain it here before we use it. So first of all, I'll create a directory which is called 16sdb, and then I'm going to also set these variables. Okay, so I will have to set the KRAKEN2db name, and I also have to set some threads here, which is this one here. So I have to set these two variables, and after that I'm going to run the script. So this is also part of the KRAKEN2 scripts. Okay, so just make sure you have installed KRAKEN2 and you have access to this script. So that's what we have here. So once you set the variables, then you have to issue this command, this script. So call the script, and then the database will be downloaded and then set up for you. So that's what we are going to do. So I'll just copy everything here. You can also download the script and use, but I'll just go and copy. I'll just copy it and then paste it. So I'll go back to the terminal. And then I'm going to open a file. This file is going to be our script. I'll use nano, but you can use any text editor. So I'll say nano, and then I'll specify a name. I'll say download underscore Silva underscore 16s underscore db dot sh. You can use any name, so it's fine. So just use any name that you feel is okay. And then I'll open it. I'll then paste what I copied into it. So I have that done. So I'll exit and save changes. I'll do an ls. And then I should see a file with the name that we specified. Okay, so this is the file. So I'll now add execution right. So I'll say chmod plus x, and then I'll specify the file name. So now we can execute the script. So let's execute the scripts to download and set up the 16s database. So I'll execute the script now. So let's wait for everything to be done for us. Okay, the database has been set up for us, and that is why we have these statements here. So let's take a look at what we have generated. So let's do an ls, and we are going to find a directory called 16s underscore db. This is where the database files have been stored. So let's ls into it. So ls 16s underscore db, and this will give us the contents. So we have everything here. So now we are ready. We can now assign taxonomic labels to our weights. So I have prepared a script for this activity also. So let's go to the GitHub repo and look at the scripts. So on this page, just search for classify weight.sh, this file here, and then click it. When you click it, the command will be displayed for you. So I'll explain a couple of things here. This command will create a directory called KRAKEN reports. So this directory is where the reports will be stored or saved. And we have also defined the database that we are going to use. That is the 16sdb. And then we have the commands here. So there are 4 commands, one for each file. So I have KRAKEN2. I also have use names, which is an option that we use to specify scientific names. And we also have the db, which is here. It's used to specify database. We have threads, which is 4. You can increase this number. There's also this option here, confidence 0.1. This one, I indicated it because the Gallery tutorial stated it here, confidence 0.1. And then I also specify the output file for the reports. That is here. So we are saving the reports in the KRAKEN underscore reports directory, the one that we created here. And we also have gzip compressed. So this here tells KRAKEN that the input file we are using is compressed. Okay, we are using fastq.gc. This is compressed, so that is why we have this flag here. So that's it, and then we save the output to the KRAKEN report here. So for each file, we are going to generate two outputs. We have the reports, and we also have this main file here, the main KRAKEN report file, which is here. So that's what you need to know. So it's the same options that have been used for the other files. So that's about it. So now I'm going to copy the commands here and then paste in another file. You can download the script also and then use it, but I'll copy and then paste the command. So I'll copy everything and then I'll go to the terminal. So on the terminal, I will open a file. So I'll say nano classify reads.sh, and then I'm going to paste the commands there. Okay, so I have pasted the commands here. So I'll exit and save changes. And then I will add execution rights. So I'll say chmod +s, and then I'll say classify reads.sh. I'll do an ls just to check the file, and that is here. So I'm now going to execute the script. So I'll say /classify reads.sh. Now with the execution, you can do it this way, or you can also specify bash. You can also say bash and then just specify the script name, but I prefer to do it this way. So that's it. So I'll run this script. Okay, so we can see that KRAKEN is doing its work. Okay, so everything has been done. So we have the summary for each file. So this one, we have 83.66% of the sequences have been classified. And then unclassified is 16.34%. So you can also check for the other ones. So what we see is that majority of the sequences have been classified. So that is okay. So let's check the outputs. So the outputs can be found here. If you do an ls, you find this directory called KRAKEN_reports. So this has the reports. So you can say ls KRAKEN_reports, and we are going to find the files there. Okay, so we have 8 files, 2 for each sample. So that's what we have. Okay, we are now going to visualize the taxonomic classification reports using krona. We have 4 reports here, so those are the reports that we are going to visualize. Okay, so the files that have the extension .reports, those are the reports. A brief description of krona has been given on the Galaxy tutorial page, so let's go and take a look at that description. So on the Galaxy page, just scroll down to this section, visualize the taxonomical classification with krona. So this is the description. It says krona allows hierarchical data to be explored with zoom-in multi-layered pie charts. With this tool, we can easily visualize the composition of the bacterial communities and compare how the populations of microorganisms are modified according to the conditions of the environment. We are dealing with a metagenomic data, and so ultimately one of the things that we would like to find out is the composition of the bacterial communities, and that is what krona helps us to do. Okay, so krona will generate a pie chart, and then we can use the pie chart to easily interpret the data. Okay, so that's one of the advantages of visualization. So visualization, and in this case, krona giving us the pie chart, allows us to interpret the data and also better understand the data. So we are going to call krona, we are going to specify the data, and we are also going to provide a name for the basal rank. We are going to use bacteria. By default, krona uses the word roots, but we are going to use bacteria because we want to follow this tutorial. So we are going to generate a plot like this, and then we are going to discuss it. So let's go back to the terminal and then continue. Okay, so on the terminal, I will first create a directory. So I'll say make the krona_reports. This is where I will save the output file. I'm now going to call krona to generate the plots. So I'll say kt import taxonomy, and then I'll say -n, and then I'll say bacteria. This is the name for the basal rank. And then I'm also going to specify -o, that is the output file. So I'll say krona underscore report slash krona.html. So this is the name of the file. Okay, and we are going to store it in this directory, krona underscore reports. We are now going to specify the reports. Okay, so we are going to visualize all these reports, the 4 reports here. So I'll say KRAKEN underscore reports slash star dot reports. And then I'm going to execute this command. So let's do that. Okay, krona has generated the plots. But if you take a look at the screen, there is a warning message here. This tells us that some taxonomy IDs were not found in a local database that we set, and because of that, those sequences have been placed in the roots. So the roots here refers to bacteria because we named the root as bacteria. Now, this issue has been reported a couple of times. I've seen a number of GitHub messages that have reported this issue, and they have provided some solutions, and I will leave the link to some of these pages in the description box, so you can use that to try and then resolve this issue. This tutorial is an introduction one, so I wouldn't go into details with how to resolve them, but for those who may encounter it, just check the links and then you'll find solutions that have been proposed. So I will proceed. So I will check the plot. So I'll do an ls into the krona directory, the krona report directory, which is here, krona underscore reports, and we are going to find this HTML file there. So this file is what we are going to view using our browser. So let's view this file, or let's open this file using our browser. So I am going to open it. It's here. The report is here. So this is it. So I'm going to open it using my browser. Okay, we have opened a krona plot. If you take a look at the left side of the page, you'll find a list of samples. There are 4 of them. So when you click on any of the samples, its associated plot will be displayed for you here. If you compare the plots we have here to the ones that have been shown here on the Gallery tutorial page, there's a huge difference, and this could be due to a number of reasons. One of the reasons would be the situation where we had some of the taxonomic IDs not being found in the local database that we set for krona. So this could be one of the reasons why we did not get the same plot as what has been given here. So in other words, the reproducibility has been affected. Okay. Another reason could be due to software versions. Different versions of the same software sometimes behave slightly different. So when we have such a situation, it can affect the reproducibility of a workflow. Nevertheless, we were able to generate the krona plots. The purpose of this tutorial was to show you how to assign taxonomic classifications and visualize the results, and we have achieved that, so that is good. Now I will switch to the original Gallery tutorial page here, and then we are going to discuss the plot that has been given here. Now, if you are going to interpret krona charts like this, then you need to also factor in certain things. Okay, for example, you need to understand the nature of the organism. You need to know the nature of the organism. When I say that, I mean you need to know or have an idea of factors that are necessary for the organism's survival and then growth and then reproduction. You also need to factor in factors that are responsible for inhibiting the organism's growth or factors that are responsible for killing the organisms. And you also need to factor in what is being done or what activities are being done in that environment, or what treatment has been given to the environment, or in this case, we have the soil. Okay, so once you get all these things in place, then it becomes easier for you to interpret. So if you take a look at the question, it says, what can we say about the health status of the soil samples? Okay, and then it also says, are there significant differences in the bacterial population structure between soil samples and rhizosphere samples? So let's take a look at the answer. So for question 1, it says the low presence of alpha proteobacteria, including members of this, as well as the abundance of this and this, suggests that the soil is highly exposed to phosphorus. So this indicates that probably phosphorus, the presence of phosphorus leads to the decline of this particular bacteria groups. Okay, and then phosphorus is also essential. It's necessary for these organisms. Okay, so we have two things here. This same phosphorus is detrimental to this group, and then it actually helps this group. Okay, so the presence is leading to the low abundance or low presence of this group, and then it's helping this group to survive, grow, and then reproduce. Okay, this mineral phosphorus is strongly associated with the anthropogenic activity, as it is an important component of agrochemicals. So this here also indicates that the environment where the soil was taken, okay, where the soil was taken from, the soil sample was taken from, probably you have agricultural activities going on there. So because there is agricultural activities, we have agrochemicals being produced there. So we have lots of phosphorus, and then this phosphorus helps this group to grow. At the same time, it's detrimental to this group here. So I need to think of this along that line. Now let's take a look at the second question. It says, are there significant differences in the bacterial population structure between soil samples and rhizosphere samples? And the answer is that we can observe that there are important differences in the structure of the bacterial population between the soil and rhizosphere samples. Particularly significant is the increase in phylum Planktomycetes, which are usually abundant in the rhizosphere. So here we are being asked whether from this chart, from the results here, whether there are significant differences in the bacterial population structure between soil samples and rhizosphere samples. So with the data that we use, we had two files with the name bulk. So we had bulk top, bulk bottom. And we also had two files with the name rhizos. That is rhizos top and a rhizos bottom. So these are the samples. So the bulk, the two files with the name bulk, those are the soil samples, and the two files with the rhizos are the rhizosphere samples. So we are basically, the question being asked is that if you compare those two samples, are there significant differences in the bacterial population structure? That's the question. And the answer is that yes, there are differences. Okay. And one of the groups that stood out was the organisms in the phylum Planktomycetes. That is what this answer is basically about. So that means that if you take a look at the plot here for the individual plots for all the samples here, and when we take a look at the plot, we will find out, or we are going to see that significant differences. So that's what we are going to do. We are going to just confirm what has been written here. The plots are here. So all that we are going to do is to look at the result that we're given, because the answer here is based on this plot. But it's important we also study this plot so that we can also agree on what has been given here. Okay, the answer that has been given here. So let's take a look at the plot. We will begin with the rhizos samples. So we have two of them, top and bottom. So let's start with the top. So if you click on the top, you will have this plot being shown for you. Okay, so here we are going to look at the phylum Planktomycetes. So if you look at the chart here, wherever you see P underscore, then the P means phylum. So that's what we are going to look at. So if you take a look at this plot, just follow my cursor, and then you will see this name here. So this is P Planktomycetes. That is what we are going to study here. That's the group is of interest to us here. So when you hover your mouse, you will have the name being highlighted. So when you see this, you can just click it. Okay, But before you click, you can look at what has been given here. So this is D bacteria. This is the total. We have 1316. So when you are here, just click this one, this particular name here. And then when you click it, you have this line being drawn, okay, to show you the regions that belong to this particular phylum. And then when you come to the top right here, you will see the percentage. So here we say, so here we see 8% of D underscore bacteria. So everything here is bacteria, but then 8% of it, okay, the phylum Planktomycetes, the organisms belong to this phylum. They make up 8% of the entire bacterial population structure, which is everything here. That is what has been given here. That's 8%. Okay, so that's what we have here. So that's what we have for this particular rhizostop sample. Now let's take a look at the rhizos bottom. But before I even do that, you will notice the color here. So there's the color for the P. Planktomycetes. So you have this color here, and you also have some here. Okay, so basically once you click this, you have this arrow telling you the groups that belong to this phylum. So take note of that. Now let's take a look at the rhizos bottom. So you can click it, and when you click it, a new chart will be displayed for you. So again, because you have already clicked the Planktomycetes for the first one, when you click on this second one, the percentage will automatically be shown for you here. That's what you have. So in this sample, the members here, the members for this phylum, Planktomycetes, they form 6% of the bacteria population structure here. That's what this simply means. Okay, so we have 8% for the rhizostop, and we have 6% for the rhizos bottom. But if you don't see the value here, let's just click it. Let's undo it. So if you click it and you don't see any value here, all that I have to do is just come here, search for this phylum, the phylum that we are talking about, this one, and then just click it. So when you click it, the percentage will be given to you here at the top right here. So you can just look at it. And if you want to remove this value or this annotation, you can just click it back and then it will be gone. So that's what you do. So we have 8% for the rhizostop, 6% for rhizos bottom. Now let's take a look at the percentage in the soil samples. We have bulk top and then bulk bottom. So let's start with the bulk top. So click it, and then when you click it, just scroll down again, and then you will look at the annotation. So when you see the annotation here, when you look at the annotation here, you will see the phylum Planktomycetes here. Okay, you will see because it also has this color. So here we can see that it forms, it makes up 0.8% of the bacteria population structure, which is small, okay, compared to the rhizosphere sample. So this is for bulk top. If you click the bulk bottom, you will find that the value has been changed. We will now have 0.6%. So this is what this question is talking about. Okay, particularly significant is the increase in phylum Planktomycetes, which are usually abundant in the rhizosphere. So here we know that this phylum, the members in this phylum, they are abundant in the rhizosphere, and that is what we are seeing in this chart. So if you compare, when you look at the rhizosphere samples, this and then this, we have 8% for top, 6% for bottom. But when we come to the soil samples, we have 0.8% for the top, we can see it here, and then 0.6% for the bottom. Okay, that's 0.6%. That's what we have. So this is what this question and then the answer are about. So just taking note of that. So in this video, you have been able to analyze 16S metagenomic data, which is a very good experience for you. So I will encourage you to also read the notes that has been given in this particular page. Okay, it will help you to better understand the 16S metagenomics data analysis workflow. So try and then read the notes here, and you'll be fine. So once you are done with this video, I'll also encourage you to watch my other metagenomics tutorial videos, which are available in this playlist.